And it's not so much that our therapies, even for cancer, are that bad. As much as you hear about that, oh, our therapies are ineffective, they're ineffective when applied late. When they're applied early, if we could know the disease existed early, the entire equation for the progression of that disease, in this case cancer, changes. So this is a very important motivating issue, something that makes me very angry personally. But that anger motivates me to do something about how we're going to take care of our children in the future and our grandchildren. So let's look at this first in terms of the value of earlier cancer detection. Shown here is a graph in which you have the stage of the cancer. So stage 1, where it's relatively localized. Stage 2, as it evolves to becoming more regional. And then, of course, as you progress to stage 4, where the cancer is spread throughout your body. And this is the survival out at, for example, 10 years, depending on at what stage you caught the cancer. This is an approximate graph. Some cancers behave a little differently. Other cancers are more or less like this graph. But you can see that when you catch cancer in this early stage, whether it's the precancerous stage or what we're calling stage 1, the outcome on average is very, very different than when you catch cancer late. And then, of course, it's a dismal outcome. And it's quite often some of the same therapies that are being used. But those therapies have a chance to work because you aren't overwhelmed with the disease in your body. Here's a PET scan of a person that has had, obviously, a lot of spread of their cancer. All these black areas you're seeing are cancer all over this person's body. This is a stage 4 cancer. This is my wife's cancer, okay, 11 years ago. At that time, because drugs were coming out that initially were only able to be utilized for late-stage breast cancer, but we said, no, we want them applied for her cancer, which was relatively early stage. You can see very intense signal from her cancer cells, and I'll explain how that signal occurs in a little bit. But you can see it's not spread like this. And of course, Aruna is alive and doing well now, almost 12 years out. So this is a very powerful issue and one to keep in mind. We cannot be satisfied with the status quo. We cannot just say, well, let's keep improving our drugs. Let's keep looking for better ways to treat. We have to do something more about detecting disease and, of course, eventually preventing it entirely. In the US and in the entire world, we spend about 100 times more out here than we do out here. This is true with National Institutes of Health funding. This is true with industry funding. This is true even with academic funding, community funding. Everyone's focused on things here because you don't think about cancer till you have it, of course. You don't think about most diseases till after you have something. It's very hard to get people to think about something when you don't feel you're affected by it. But of course, many people in this room already harbor a cancer, one that will grow in some cases over time and be aggressive, and in other cases not go on to do any harm at all. And so it's not even about finding cancer early, but it's about finding aggressive cancer, the kind of cancer that will go on to hurt a given individual, to kill a given individual. So many things are changing that are going to allow us to do a better job. Certainly here at Stanford, with efforts in the Department of Genetics, the Department of Pathology, we are seeing a revolution in how we can start to understand what are problems in your genetic code that might make you susceptible to certain kinds of cancer. Simple blood tests taken eventually at the time you're born, but right now, taken now, let us quickly profile your gene sequence and arrive at errors that tell us that you might be susceptible to a certain kind of cancer. And so it's not just about necessarily screening everyone, but it's about screening those individuals that you know are at risk. Eventually, of course, as our tests get better and better, they can be applied to everyone. But initially, the tests will be applied to people at risk for certain kinds of diseases, including cancer. So the motto we've been preaching for quite some time now is these 3 I's: identify, image, and then intervene. We want to identify disease very early with low-cost tests that are very accurate. We don't want to put everyone through expensive imaging tests if we can avoid it. We want to develop lower-cost imaging tests, but more likely than not, some form of blood or other bodily fluids like urine or saliva or even your exhaled breath will be combined with imaging to let us detect the presence of the cancer and then let us localize it with imaging. The blood test can't tell us where the cancer is exactly, so the imaging is still needed to localize it. And then we can start to intervene at very early time points. Time points when, for example, a cancer might be just a couple of millimeters in size. And that leads us to many options for intervention, including intervention with things like sound waves that can just heat up the tumor and destroy it, or looking at technologies where there are targeted drugs, but you open up many, many more possibilities once you can intervene at a very early time because the cancer is still very, very small. Why is this such a difficult problem? Why is it that it hasn't been already done? Well, when you talk about a cubic centimeter of a tumor, about a ½ inch on side, that's already 3 billion cells. So you know, when my wife, for example, felt a mass in her breast, that mass is already several billion cells. Okay, and of course it's these cells that have gone awry that are rapidly dividing and are the ones that will go on to spread to other parts of your body to eventually kill you. Well, if we could come down to even a cubic millimeter, we'd still be at 3 million cells. And of course, if we can come down to 0.1 cubic millimeters, not 0.1 on each side, but just 0.1 overall, then it's about 300,000 cells. And it's very hard to push down this low, because how do you find so few cells? If they're deep somewhere in your body, how do you know where they are? And the fewer they are in number, it's like trying to find a very, very small signal that could be hidden. And of course, if the tumor got larger and larger, it's easier because there's a lot more signal, and I'll tell you what I mean by signal. And so that it's a difficult challenge. But here at Stanford, working with the School of Engineering, we are setting our sights on approaching things like this, things that people think right now are impossible. Well, one way to do this is based on sampling things in your blood. Cancer cells, for example, do in fact die. Some die as they divide naturally. Some shed markers into the blood. Those then can be found in the blood potentially. These are proteins that are coming from cancer cells, DNA, RNA, microRNA, even small molecules. There's now a lot of studies about things in your breath that are being exhaled from you that we didn't think were there, that actually tell us a little bit about the fact that you might have hidden disease. And then, at least in later stage cancer, some of the cancer cells shed off and enter the bloodstream because that's how they spread. And so circulating tumor cells, as they're called, are another thing that's a possibility. But many of us feel those might be already too late. We don't want to catch things by the time tumor cells have entered the blood. We want to know well ahead of that. So the blood sampling, or urine sampling in some case, has potential. But it's tough. It's tough finding very, very tiny amounts of things circulating in your blood. We've been mathematically modeling this to try to put a mathematical framework around the problem, to say, how do we know if things are being shed from cancer cells and also potentially from normal cells? How do we know whether we can even hope to ever find a tumor that's just a millimeter in size or 0.1 millimeters in size? Maybe it's an unrealistic goal. Well, using mathematical models and understanding the rate at which tumor cells shed certain proteins, for example, and the fact that normal cells also might make tiny amounts of those proteins, we can model all this, and I won't bore you with the mathematical models, unless you want to be bored. But the idea is that we have shown that we can cross through under a cubic millimeter. And we know what kinds of technologies we have to build in order to cross through those barriers, and we know what kind of proteins have to be present in the blood for us to be able to detect tumors that may be hidden in your body. So we have set out over the last few years to build technologies here at Stanford that can, first of all, pull off this goal of detecting these very, very low levels of telltale signs in your blood. How? How will we do this? Well, this is where we go over to the School of Engineering. The strengths at Stanford are not just a medical school sitting by itself, but an engineering school, a humanities and sciences school, which is where chemistry and physics are, and of course many other schools that we also take advantage of. And we start to work with our collaborators in those schools. And we set a very high bar. We said we want to detect extremely low levels of proteins in the blood, something known as femtomolar, 10 to the minus 15th molar. It's a tiny amount. 6 log orders of dynamic range, meaning we want to be able to detect low levels of proteins and high levels of proteins. We want to be able to detect many kinds of proteins, up to 64, coming from tumors. So we don't want to just look for one thing coming from tumor. We want to sniff out many things that tumors are making. And we also not just want to look at blood, but we want to be able to take saliva. We want to be able to take urine. It has to be low cost. We want these technologies eventually to be $5, $10. We want it to be a home test. You could prick your finger, drop the blood, just like you would for blood glucose, and have the chip tell you the answer. Well, you're looking at the first generation of such a chip. This is known as the Magnetonanosensor. It is based on technologies that gave birth here in Silicon Valley, the same thing that's in your hard disk drives, the giant magnetoresistor, which helps the hard disk drive in terms of storing information. We harness the power of the GMR in these chips. And the way these chips work is the following. We have this little sensor shown here in yellow that only senses changes in magnetic field. It doesn't know anything about cancer. It doesn't know how to recognize something in your blood that's from cancer or not. It just knows about changes in magnetic field. But the tiniest changes in magnetic field it can detect. We put on this sensor little proteins, antibodies, that are designed to lock in to anything floating around in your blood, even if there's a very tiny amount of it. This one might lock in on one thing being made by a tumor. This one might lock in on another thing being made by a tumor, and so on, up to 64 of these in the current chips. Now we drop your blood on there, and if your blood happens to have tiny, tiny amounts of a protein that's being made by a cancer cell, this locks onto it. The sensor still doesn't know anything's happened. There's no change in magnetic field. The sensor says, well, nothing's going on. We come in with another molecule with a tiny little magnetic nanoparticle. You hear about all the hype of nanotechnology? Well, this is very real applications of nanotechnology. This little magnetic nanoparticle, which is attached to this, which is attached to this, which is attached to this, causes a change in magnetic field. And now we know there's a change in magnetic field at this location of the chip. And therefore we know, because we know what we put at that location of the chip, that there must be something in the blood that's locked on to that location. This technology has been in evolution for about 7 years now. It's been commercialized as MagArray. It's a startup that Toyota has actually invested in because of their interest in magnetic-based technologies. It's read out with a simple little reader. The chip goes in and it's read out. And to show you that this is very real, Eric's going to actually pass out the chip to you. You can touch it. Don't put your drop of blood on it. But do look through it because it is quite real. So these things aren't magic workers. They don't know what to look for in the blood. It's not like the chip can figure out everything. We have to functionalize it. We have to train it what to look for in the blood. But once it does, it outperforms anything that we have an understanding of right now. A blood test in a hospital, which is called an ELISA, when someone orders your blood test for a given marker in your blood, its sensitivity, that is how low a level it can detect, is around here. But this chip crosses over into an extremely, extremely sensitive level. So very few cancer cells making very few levels of these proteins have a chance of being detected. And it's a perfect example of something that no other university can do as well. This isn't a chip that exists if you went to Harvard or exists at Caltech or UCLA or UCSF. This is something born here at Stanford and the strength of this great institution and schools working together to solve problems in human health. When we apply these things in mice, these are 5 different mice where tumors are growing. We can see in the mice's blood a protein marker called CEA, which is increasing in their blood. If we applied current hospital tests for the same marker, the hospital test would say there's nothing in the blood. It would say there's no cancer, there's nothing growing at all. We have crossed into a range that we never thought would be possible to see. And that is opening up now the chance to see things that we never thought were there even in patients. For example, women have had archived away their blood each and every year for the last 10 years because when they were getting their routine screening mammograms, their blood was collected and the blood was stored away. But then some of those women go on to get breast cancer, some go on to get ovarian cancer, and we have their blood. It's very precious because everyone wants that blood to know what's hidden in that blood 10 years ago that is now potentially going to reveal that they're going to get cancer. Because if we try to do the study now, it'll take us 10 years from now to figure it out. Well, luckily, actually one group in Canada has this stored blood. So we are testing right now in a large-scale clinical trial proteins that are potentially indicative of early cancer. And it's not just the presence of cancer, it's how aggressive it is. We want to know, is the cancer not just there, but is it making proteins that tell us that it's an aggressive cancer? Because those are the kind that we want to intervene on. So this study is currently ongoing between Stanford and the Fred Hutch Cancer Center in Seattle, and we will hopefully see what it reveals for ovarian cancer. Then we're asking even more unusual questions. We're saying, but even if tumors are busy making these biomarkers, we don't want to wait and bring a person back 6 months from now, a year from now, and see, well, did their blood levels of the protein go up so that the cancer is likely there? We want to kind of stress the tumor. We want to yell at the tumor and force it to reveal itself, okay? How do we yell at it and force it to reveal itself? And we're literally yelling at tumors. How do we do that? If cancer cells exist in your body and they're busy making proteins, in this case, these little green balls that are entering the blood, what if we said we could use sound, and using sound, if the sound hits the right part of the body and a cancer is hidden there, then all of a sudden the cancer starts to shed more balls. And when we first proposed this, people said, this will never work, it's crazy. Why would sound even cause cancer cells to get scared? Well, as it turns out, sound at at the right frequency, is able to increase in a very transient way, in a very small way, the ability of these proteins to be shed or increase in the blood. And so this clinical trial has also started. Imagine that you have patients at risk for ovarian cancer or breast cancer or otherwise. If you took their blood and they never had any cancer at all, you wouldn't see, hopefully, much of those green balls in their blood. But if you put sound waves in the area of the ovaries, in this case, in this patient's right ovary, but there's no hidden cancer there because they don't have a cancer, then still nothing happens in the blood. But if there's a hidden cancer there and you apply sound, now these cells shed those biomarkers in the blood and all of a sudden they're detected. So this is literally like a stress test for cancer. You might say, but I don't know, where are you going to apply the sound? Yes, but we know what you're at risk for. If you're at risk for breast cancer, we know where to go. If you're at risk for ovarian cancer, we need to go. But we don't have a way of applying sound to your whole body, in little array-like fashion. We have to pick a site. So this clinical trial is also now started here at Stanford to see if we can, in fact, try to pick up early signs of cancer. Okay, let's change gears. Lots happening in this area. This in itself is an hour talk, but let me shift gears to imaging now. We, of course, want to develop the penultimate scanner. We want to be able to have you standing, maybe even in your shower someday, and be being scanned, okay, without you even knowing it, unless you wanted to not share your information. But the scanner we want to develop literally, can scan your entire body and produce beautiful pictures of the anatomy of your body, the molecular signals within your body, do it at very high resolution so that we can see things. Now, the blood might have told us you have something, but the scanner tells us where it is. And we're building these molecular spies that go into your body and hunt down cancer. They're literally spies that are injected into your blood. They wander around, do a house-to-house search, a cell-to-cell search, looking for cancer. And we test these in small animal models and then move them into the clinic. There's actually such a molecule now. If you were in the last talk by Frank Longo, there's such a molecule for early Alzheimer's disease that's just gotten FDA approval. But we build many of these molecules that are designed to look for certain irregularities in cancer. This is the one you would see in the clinic right now called fluorodeoxyglucose. It basically is a sugar molecule, and cancer cells love to eat sugar. And so they take up more of this than your normal cells. And then we have a little walkie-talkie on the molecule. We literally have the molecule send us signals so that we can know where the molecule is being consumed in the body and therefore can localize the site of the cancer. As a matter of fact, my wife's scan that you saw from about 12 years ago was this molecule at work in a PET scanner. It was what lit up that breast cancer you saw. But we're building a whole new cadre of new kinds of imaging strategies. For example, now we're building bubbles that are targeted with things on their surface that help them go find abnormal cancer cells and abnormal cancer blood vessels. These bubbles are filled with a gas. The gas allows the bubbles to oscillate using an ultrasound machine, and now you can start to pick up cancer cells hidden in that tumor area that you might have suspected. We're also building all kinds of miniaturized microscopes. Dr. Contag, other groups here at Stanford, optical engineering at Stanford, are literally working on microscopes that go into your body and let us look for, with little spies that light up bad areas, in this case, gastrointestinal cancer, colon cancer. So microscopes will start to go into your body. This always makes my colleague Steve Galley worry because he works in the Department of Pathology. And his goal is to make our department obsolete. My goal is to make his department obsolete. So he's in the back there smiling, sweating right now. But he knows that the goal eventually is to do whatever pathologists do now, but they have to remove tissue. But we want to do that while the tissue's still in someone. And hopefully the next generation will allow that. Another example of a great technology developed here at Stanford, photoacoustics. This is where light goes into the body and sound comes out. Right now, all of you are actually making sound. Listen. Okay, no one's hearing it. Okay, unless someone's doing an unusual kind of sound. But this light that is going into your body is actually heating up slightly molecules, including molecules in cancer cells. And that absorption of light leads to the heating, which leads to thermal expansion. It's not much heat. It's like 0.1 degrees. But the point is, we pulse laser light in, and out we get is sound. This opens up all kinds of ways to see things in your body and send in little spies that are good at heating up and good at absorbing light. But they also are good at finding cancer. And this is one kind of spy that's based on carbon that basically can go into the body, and as it goes into the body and as it's trained to hunt down cancer blood vessels, it actually lights up where those are. And it's now sound coming out of your body that's telling us where things are. So for example, in the future, instead of a mammogram, which is good in its own rights, but involves x-rays, still can't have the accuracy we'd like, there's many false positives, will change because we'll pulse light in, as we did here in this human breast. Light is entering the top of this human breast from a laser, and while you're seeing what looks like blood vessels throughout this patient's breast, is the hemoglobin in the blood is absorbing light, slightly heating up and producing sound. And that's letting us see the entire breast in 30 seconds at a spatial resolution that's unimaginable, 200 microns, and all with technologies that are extremely low cost. This whole system would be $70,000, where the equivalent image that would take about 30 minutes in an MRI camera would be several $million. So we're paying a lot of attention to not just technologies we can build and deploy in our community, but also throughout the world. There is a huge explosion of these regional technologies. We can look for disease in your esophagus, disease in your bowel, disease in your breast, in your ovaries, in your lungs. We don't have that whole body scanner yet that can detect at this level disease anywhere, but we're solving it regionally with regional imaging equipment. So finally, to end, there's a huge promise of things coming that will marry these two fields together. We believe then that intervention will fundamentally change. It will be possible to intervene when you have a cancer that's a few millimeters in size or a few sub-millimeter in size, and then totally change the outcome for you as an individual, for your children, and for your grandchildren. So I've shown you examples of technologies all being developed here. Blood testing, in vitro, so-called testing, blood testing, urine testing, being combined with imaging, so-called in vivo testing. These are coming together to allow earlier detection of cancer. As technologies to solve the problem of early cancer detection, in fact, are validated, they will also lead to other benefits. If we can solve the problem of seeing so few tumor cells, make no mistake about it, that means we will also be able to markedly help when you have more disease because you weren't screened for some reason. So this is the hardest problem, and it will make the other problems that much easier. The technologies are ahead of the biology. We still don't fully understand all the things that make cancer cells different than normal cells. A lot of these technologies can't work miracles. Biology's had a long, long time to evolve, and we're at the infancy of understanding it and marrying it to the technology. So don't feel that technologies can solve it all alone. They need investments in the biology to solve. And finally, we're very conscious about validating things clinically, overdiagnosis. We don't want to be diagnosing everyone with cancer, cancers that won't go on to hurt you at all, only cause anxiety. The health economics of all this, and of course, patient compliance. How do you get everyone to actually do all this? We recently expanded with the Canary Center at Stanford, which is focused on early cancer detection. This is a huge investment by the community and by Stanford Hospital to build the next generation of tools. It's on California Avenue, and we'll move to the new Technology and Innovation Campus on Porter Avenue. This is the first place in the world where the in vitro world and the in vivo world are coming together. We are also investing a lot in the hospital with next generation imaging technologies, things that are being developed here on campus that will move into patient use for the first time. It takes a lot of different kinds of scientists to bring this work together. Chemists to make the molecular spies, proteomics scientists that study proteins, physicists and engineers that build instruments, material scientists that build nanotechnologies. Massive effort on the part of many different people, and I'm just up here representing some of them today. And I hope I've shared with you in this brief 30 minutes what may be possible in the years to come, and I hope you're just as excited about it as I am. Thank you very much.
In dermatology, we have an advantage. It's all there to see, and we use the dermoscope a lot. But what my question is, in the prepping that you're doing with ultrasound, et cetera, and other things, what evidence do you have that this doesn't change the behavior of the cancer?
That's a good question. So in the example we give, which we yell at the tumors, the last thing you want to do is yell at them and make them angry so that they actually then go on to spread, for example. So how we look for that is we use a technique called intravital microscopy, which lets us look at the tumor through a window placed, in this case, on an animal. And a microscope lets us study the cells while we hit with sound. And then we watch to see if, for example, we're dislodging any cells, or some of the cells actually being dislodged by the sound, allowing them to spread. And no, there's not evidence of things like that with the models we've studied so far. But it's a very important issue. You certainly don't want to disseminate the disease or make it worse by perturbing it. But this sound is not, the kind of sound that you would think is destroying a region or blowing up a region or heating a region, which you can do with sound, as the military does with sound guns. This is a very special frequency which actually just opens up micropores on the cell membrane that allows things to escape from the cell, as opposed to letting the cells move. Yeah. Yes. Yeah. So I'll repeat that question. Question is, the early detection of prostate cancer hasn't really improved outcome. So what's the issue there? Well, it's interesting when you back up about a decade or so ago, of course, what was thought is, well, if you have prostate cancer, you have to act, you have to remove it, okay? And now we've learned that's not the case. Matter of fact, if any of you are diagnosed with prostate cancer now, you'll be offered what's called active surveillance, prostate active surveillance studies, which are now national studies going on, where you say, you may have a prostate cancer which is not aggressive at all. And in fact, had we not even caught it, maybe it would have never gone on to harm you. You would have died from other causes. And so we're learning the issue that it isn't about early detection of cancer. That's why you kept hearing me say aggressive cancers. The goal is not to find things that all cancers are making that might, if we deployed them, cause all of you to be diagnosed with cancer. The goal is to find things that only aggressive cancers are making. And if we don't, we end up in the problem of overdiagnosis. So in prostate cancer, one of the dilemmas has been, as we start to have blood tests, which, as good as they were in their time, didn't employ the kinds of technologies we have now, unfortunately, we're not indicative of aggressive cancer, okay? PSA, prostate-specific antigen, which is the blood test you get now, that molecule is made by healthy prostate cells and prostate cancer. It's not as specific as we would like. And also, it's not indicative of an aggressive prostate cancer. But if you had a another molecule, actually one that's been discovered recently at the University of Michigan, that's more specific for aggressive cancer, potentially changes the equation. So that's one of the reasons that's been happening. Yeah.
Wouldn't treatments have to be different if you have very early detection, or, you know, even detection before there's almost any tumor, because patients might be reluctant to get aggressive treatment when they, you know, that they're not even sure that they really will develop cancer?
Yeah, so let's say that you had an early cancer and we're not sure how aggressive it's going to be. And do you, in fact, even treat it? And would the treatment have to be different than for a more aggressive cancer or later stage cancer? Yes, possibly. For example, in the prostate case, just as an example, patients right now who have prostate cancer are being watched. The prostate cancer is not being removed. They're coming back every 6 months to 12 months, and we're doing studies to look at whether their prostate cancer is showing signs of progression, because many won't, so there's no reason to do anything. That's why it's important to be able to select out the aggressive ones from the non-aggressive. But let's say now you can't still be sure, but you've still caught something early. Could you intervene with new kinds of technologies? Yes. Right now we have a technology known as HIFU, high intensity frequency ultrasound, where we can literally dial in almost anywhere in your body a coordinate and say, destroy that area of the body. So we can say, okay, I want you at such and such GPS location within the body to take a 2 millimeter volume, heat it through sound, and destroy it. It's not radiation, sound waves. It opens up technologies that can act regionally without you having to take a drug. But we do have to be careful not to start treating everything, because, as you're rightfully stating, many things won't go on to hurt you. So that's why we have to avoid over-diagnosis and pay a lot of attention to really developing tools that catch the bad things. Yeah.
Is a PSA test irrelevant now?
I'm sorry?
PSA, is it irrelevant? You don't use it now?
There's still a debate, a strong debate about PSA. You'll hear in the literature and in the news, one group saying, no, you know, PSA is not needed. Other groups saying, no, that's a crazy statement. Generally speaking, there is a role for PSA in different arenas. But the more important thing is, just because you have an elevated PSA doesn't mean that you have an aggressive cancer. You have to watch what the PSA is doing over time. You have to combine other tests, including biopsy results, genetic analysis of your tumor. So the story's still not out. But it's not the case like we thought well over a decade ago, even here in the Department of Urology, that PSA was a definitive yes or no answer to whether you need treatment for prostate cancer. How will that change? Because we will start to have a series of PSA-like markers. One that's more indicative of aggressive prostate cancer, another that's indicative of aggressive prostate cancer, and a panel of biomarkers is likely to be more predictive of whether you've got a bad tumor versus a not so bad tumor. Yeah, sorry.
Elevation of the alpha-fetoprotein is used to monitor liver cancer, but it's not very effective because it's very delayed.
Yeah.
Is it a marker or indicator for liver cancer?
So for liver cancers, there is still a hunt for better biomarkers. Actually, for most of these cancers, you might say, well, how come that magnetonanosensor isn't available at my house today? Why can't I drop my blood on it today? And maybe it could have 64 different things it could look for, like liver cancer, breast cancer, so on. Part of the problem is that it's hard finding proteins or other markers that only cancer cells make, because most things are also made by normal cells too, because in fact your cancer cells are your cells gone awry. But now, through very advanced science in proteomics, genomics, we're discovering new biomarkers. Matter of fact, we're discovering them so quickly, we don't have the ability to test them all fast enough. Because you can't just take a biomarker, a protein like one for liver cancer, and say, okay, it's ready to go. Many, many things occur to show that it can work in a population of interest. It has enough accuracy. And so you won't see tests overnight because there's a lot of validation for these panels of proteins, panels of biomarkers before they're ready for prime time. Will they occur? Probably highly likely. What if they don't? Imaging is the second sister technology where we send in spies that go look for the cancer. And in liver cancer, that would occur because we have spies that are going and designed to find abnormal liver cancer cells. The two technologies are very much, I kind of equate it sometimes to the field of astronomy. In astronomy, you're trying to figure out what's going on at a very far distance. And astronomers are very envious of us because we can even grab the planet, if you will, and interrogate it. They can't grab the planet. But they've learned things like how to observe things coming from the planet and how to send satellites and other tools to a body. In some ways, the cancer problem's like that. We can sample the cancer by not being at it, by going to the blood or going to the urine, but we can also send things in that then go sample the cancer, if you will. Some combination of the two is what it likely takes to eventually win the war. Yeah.
You made a comment that some cancer cells love to eat sugar, and I wondered what implications of that there are for prevention and slowing progression.
Right. It's a great question. The answer is hidden in these images, but let's go through it. Originally, when it was discovered that cancers love certain kinds of sugars, like glucose molecules, people said, oh, well, let's just build things that kind of look like glucose, tricking the cancer cells, because they'll eat a lot of those, but then won't be able to metabolize those sugars and will eventually starve themselves. Okay? As a matter of fact, if you take cancer cells outside the body and feed them sugars that kind of resemble glucose but aren't glucose, they die. If you give them glucose, they keep growing like crazy. So then, a group in New York actually said, okay, let's start giving cancer patients these sugars because we'll try to kill the cancer cells by starving them of glucose. But guess what went wrong? What went wrong? Why don't we hear about things like, let's go get glucose therapy for cancer? What in your images, including in my wife's image, did you see other than that spot in the breast cancer area? Where else? Someone's pointing. The brain. You saw that the sugar was being eaten by the brain cells. Let's go back to that for a minute. Look at her picture there, right? The intense cancer right in her right breast. Our left is her right. But look at the brain. So when you try to give these sugars to a living animal, because you have to do that before you go to a human, guess what happens to the animal? It goes into a coma. Because the brain needs sugar, and you're busy starving the brain of sugar because you're giving it a mimic of sugar. So what if you say, well, but let's just not give you a mimic, let's just lower your sugar levels. Why don't we just, have you lie down and keep you hypoglycemic, keep your blood glucose low? Unfortunately, the body's designed to make sure that doesn't happen to a significant degree because the brain always needs glucose. So if you try to bring your body sugar way down, you put yourself into a coma. And so there's a huge balance between that. If the brain wasn't so dependent on sugar, you potentially would have an alternate way of killing cancer cells that way. Yeah. Yeah.
Part of that, some people have looked at ketogenic diets that lower the blood sugar. I'm sorry. Some people have looked at ketogenic diets. The brain can survive on a lower glucose level done properly. I don't know that that's proven very effective yet. What is effective is trying to keep blood sugar at least in the normal level, and we know that diabetics have higher risks of cancer. We know that obesity increases the risk of cancer, so that all the things that you're told to do for your heart and for the prior study on talk on dementia, the same thing goes for cancer. At least keep body weight and blood sugar levels good. There's also some studies being done with metformin, which as a cancer adjuvant therapy.
Yeah, there are definitely approaches being tried. The whole area of metabolomics, which we didn't talk about at all, the metabolism of cancer cells and how to take advantage of the metabolomics, including glutamate utilization, something known as the XC transporter, changing dietary patterns, all can potentially help. The problem is that in cancer, it's very difficult unless you can really control all the cells, right? Even if you slow down the growth and maybe try to make it a chronic disease, it's difficult without really hitting all the cells. And it's an important area of ongoing research, and hopefully someday that'll be answered. But like I say, I believe the answer isn't even so much that. If you can catch this cancer early, you can already intervene with what we have now. Drugs, local regional therapies, and it's a totally different outcome for most cancers. I'm going to stop with that. Thank you all very much. Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.